One of the most important techniques that you're going to have to master for your microbiology lab is to be able to make a sterile transfer of your microorganisms. An important part of this is to always start with a nice aseptic workspace, which Sarah is working on right now. When I say aseptic transfer, what I mean is that you are transferring bacteria from one tube or one plate to another, and you're doing so in a way that you can be very confident that you have just transferred the microbe that you want to transfer and not brought anything else along with it. So once you have a nice clean bench space, it's important to get your flame going so that you can have a nice sterile zone. There we go. And I always like to say that you should be working definitely within 1 foot of your Bunsen burner. And the closer the better. I like to keep it within about 6 inches myself. So once you've got your flame going, it's important to have a nice organized workspace. So any transfers that you're about to do, make sure that all of your tubes are well labeled. So you want to know what organism is going into that tube, what media is already hanging out in that tube, and also the date, and often a few other important details, but those 3 key ones: your name, name of the organism, the media, and the date. So I guess that's 4 key pieces of information. And Sarah's already done most of her labels before getting started, so those are all set and ready to go. So it may be a little bit hard to see from here, but the tubes that Sarah's got are not all the same. So some of them are liquid. So you can see if she jiggles them back and forth, that's going to splash around a little bit. So those are broth tubes. And the other type of tubes she has there are slant tubes. So I'm just going to zoom right in. You should be able to see that there is a solid media and it's on a diagonal. So that gives a nice growing surface for the bacteria. Excellent. I also want to point out how Sarah is picking up the tubes. She's always putting her fingers on the glass and not picking them up by the lid. It's often good to put a finger on top of the lid so that it doesn't slip off, but you never want to pick it up by the lid. That's a very easy way to spill your microbes. So to start with an aseptic transfer, we're going to start by transferring organisms that have been growing on an agar plate. So here we can see, you can actually see that there are lots of individual colonies on this plate, and Sarah's going to be picking from just one of those colonies. Now, it might look like that's a small colony, but it is more than enough for what you need to transfer. In fact, you're never going to take a full colony. You're just going to take a little bit out of it. Now, how are we going to move this colony? We need some sort of tool to get it from one place to another. And the tool we're going to use is an inoculating loop. So I'll zoom right in there and see, there we go. You can see that that loop actually has a circle of wire at the end. I'm a little bit too zoomed in, there we go. So we've got a little circle of wire at the end, and that will allow us to move our bacteria from one place to another. The wire is quite flexible. Every so often it gets bent. So if that happens, you just use your fingers to straighten it out. Now, since this was just touched and has just been hanging out in the lab, it is absolutely not aseptic at this point. So what needs to happen is it needs to be flamed in your Bunsen burner. So to do that, you put your flame or your loop right into the hottest part of the flame. And you should be able to see that nice glow happening there. That tells us that the loop is actually getting hot enough. And then you just slowly move from the base all the way to the tip, making sure that every part of the loop gets red hot. So that loop is nice and sterile. You want to make sure that from this point forward it stays in that little sterile circle. So no more than a foot away from that flame. And if Sarah went and did that transfer right this moment, that loop would probably burn and kill any of the bacteria in there. So she's just letting it sit for at least 15 seconds to cool off. Once it's ready, she can open the plate of bacteria. The way to do that is to leave the lid of the bacteria on the bench and to lift up the part that has the agar. Then just open it so it's facing you and gently touch your loop to one of the colonies of bacteria. Great. Now you can close the plate back up, and we're staying in our sterile zone, and we can pick up the tube that we want to transfer into. So to pick this up, we don't want to have to put anything down on the counter. So instead, Sarah's just going to use her little finger to grab that cap and just gently pull the rest of the tube, and then flame the mouth of the tube just quickly. We don't want our hand to get too hot. And then the loop can enter into the broth, go right into the liquid, and just wiggle it around and get any solid material off of your loop. And you can bring it back out, flame the mouth of your tube again, make sure none of your hand microbes got on there, and then flame your loop again so that it gets sterilized again and is ready to go. So again, right to red hot, and then set it aside to cool. So that's our first transfer. So we're going from a solid media into a liquid media. We can also transfer from a solid media onto another solid media, so onto an agar slant, like we're doing here. So that loop left our sterile zone, so it needs to be sterilized again. So we'll get it nice and red hot. And here's time for another one of my pro tips. If you want to set your loop aside, you can just keep your test tube rack nice and close to the flame, put the handle in the rack, and now your loop is staying in the sterile zone while your hands are free to do other things. So we've got, another 10 seconds or so for that loop to cool. In the meantime, let's make sure that we've got our transfer tube ready to go. We've still got our nice solid plate of E. coli there that we're ready to transfer from. And something to note is that when Sarah picks up this next colony, she's going to pick from a different colony than she already touched. So we'll go for a fresh one this time. All right, that loop should be cool. If you're not totally sure, you can just find an empty spot on the plate and touch the loop down to the agar. If it sizzles, you need to wait a little longer. If it doesn't sizzle, you're good to pick your colony. So we can go in and grab a colony off the plate, put the plate back down. Same technique when using the slant. We're going to hold the cap in one hand, flame the mouth of the flask, and this time we're going to put our loop down in, and we're just going to zigzag it back and forth on that agar slant. And then once you've reached the top, you'll just bring the loop out, flame the lip again, or the mouth of the tube, and set your tube aside. And again, get your loop nice and sterile for the next go around. So those are a couple of different transfers that we can do, taking our culture from a solid source, but we're not always using a solid source. Often we want to take from a liquid media. So in this case, this tube in Sarah's hand, you can see that it's a little bit cloudy in there. That's because it's got bacteria growing in it. So before we do anything with this tube, we want to make sure that the bacteria are evenly in the solution. So we're going to do that using this device called a vortex mixer. And to use the vortex, always make sure you've got a good grip on the glass, and definitely make sure that your finger is on the lid there. Then you can press it into that black cup, and it's going to make it swirl in a kind of tornado or vortex in there. So that's well mixed now, and we're ready that we could take a sample out of it. Now Sarah kept her loop in the sterile zone, so it is nice and cool and good to go. So she just needs to take the lid off of her tube, flame the mouth of the tube, and then she's just going to reach her loop in there and maybe wiggle it around a bit, and then flame the loop of the tube again. Now I'm not sure if I'll be able to see this. Sarah, hold the loop there. If we were looking really, really carefully, which we can't, we're not going to focus, that's okay. What I can see though is that there's actually a film of liquid in that loop. So now we know that we're transferring bacteria. We flame the mouth of our recipient tube. We put our loop right in there and just wiggle it around to kind of mix in solution there. Then exit, flame the mouth again, and sterilize our inoculating loop. So we can also take a liquid culture and transfer it onto solid media. And it's going to look exactly the same way as taking it from a solid and putting it onto a solid. But let's just run through it one more time so that you get to see that practice. So that loop has been sterilized. It's going to be cool. We want to vortex our sample again, make sure it's well mixed. Then same technique, lid comes off. Mouth of the tube gets flamed. Loop gets dipped in the culture. Mouth gets flamed. And the loop full of culture gets transferred over to our tube. Flame the mouth first, and this time we're just going to zigzag our loop up the surface of the slant. And then exit, flame again, and cap back on. So all of the transfer techniques that we've shown you so far are a great way to be able to just transfer some bacteria from one place to another. Sometimes though we want to go a step further and actually be able to separate individual colonies from one another. And for that we'll use a technique called the streak plate, and we'll be going through that in our next video. All right, we'll see you back soon. So we're here to demonstrate how to do a streak plate. And yes, Sarah's over at the whiteboard instead of at the bench, because the most important first step of doing a streak plate is making sure that your plate is properly labeled. So you always want to label your plate around the edges of it, and you want to include the media, usually that's TSA, the organism that you're transferring, we're doing E. coli today, the date, and then also your initials. And remember that this label is going on the side of the plate that already has the agar media in it. When you're doing a streak plate, you've already gone through the basics of this in your lecture, and we talked about how we use specific quadrants to do a streak plate, and we really recommend that you draw out your first quadrant. So you just draw a line that shows you where you're going to fill in your bacteria. And when you actually start to transfer your bacteria, you're going to completely fill that line in. So that's going to be, end up becoming completely solid, and then you'll move on from there. I actually like having it in the same area where your label is, because then all of your isolated colonies are going to be far away from any markings that are on your plate. So that's the area that you'll just go back and forth, back and forth. You can make it all the way to the edges if you want to. Sarah doesn't need to demonstrate that though with the marker. You get the point. You're just moving your loop back and forth, back and forth, back and forth. Then you will flame your loop and go all the way to the one edge of your plate and drag your loop across. And then you can either pick up and go back like Sarah's doing here. Some of you might like zigzagging back and forth. That's an option too. Either one is okay. But you make sure that it only crosses that first zone twice. And then the rest of your transfers are all going to be on the blank, empty portion of the plate. And you're going to fill in that next section about like that. You want to make sure that you've still got enough room for your third quadrant. Then you flame your loop again and start from that edge, drag it through all those lines, drag it through all those lines again, and then do not touch the first zone and just wipe your loop on the clean, untouched section of agar. This way, we should be able to get growth of individual bacterial colonies. So that's how this works in a whiteboard type setting. And now Sarah will come back to her workspace and show us how it works when you're actually working with bacteria. So Sarah has already labeled her plate and got all the important information on there. The one thing that's missing is her little line separating the quadrant out. So there we go. So that's going to be the area that gets filled in, all that area above that line. So now that everything is labeled, and of course Sarah already cleaned her bench space before getting started, now she's ready to get her space organized, make sure that the loop's nice and close, make sure that you've got a good working area with the Bunsen burner, and we can get that burner lit and good to go. Perfect. So just like with any other aseptic transfer, your first step is going to be to flame that loop, get it nice and red hot. There we go. And then give that a moment to cool. I'm going to be honest with you guys, every so often I cheat a little bit when I'm using solid media, and even when my loop might not be cool, sometimes I'll touch it to the media anyways because that helps to cool it down a little quicker. Just make sure that you're nowhere near your colonies when you're doing that. So now you can, just like when you were doing your transfer into tubes, you'll touch a colony. You can actually pick up enough that it's actually visible onto your loop, but you don't need to pick up the entire colony. And then you can move over to your clean plate and fill in your zone that you've drawn out there. And Sarah's just going back and forth with her loop, filling in that area. You can go back and forth a bunch of times, then you've put your plate back down onto the lid and you flame your loop and let it cool. Now the loop is a little bit too thin for you to have been able to see this, but when Sarah was spreading with her loop, for that first quadrant she was putting it so that the entire face of the loop was up against the agar surface. So you're trying to transfer as much bacteria as possible in that first step. In the future steps, you only want the edge of the loop to touch down onto the agar. So it's like the circle is up and down instead of being flat onto the agar. So now Sarah's going to open up, her loop is cool. She's going to start in that first quadrant, drag it out, do 2 lines like that, and then continue in the blank space there. Then the plate can go back down, loop gets flamed. And the loop gets a chance to cool. And then we'll just have that last quadrant to go, and really we're doing it in thirds, not quadrants, but some people do it in quadrants, that's perfectly good too. 3 is usually enough for the type of bacteria that we're working with in this lab. So now that that loop's had a chance to cool, we can open up our plate again, draw through those initial lines, and continue to wipe it out onto the plate. And then we just flame our loop, and our streak plate is ready to be incubated. We'll walk you through getting your plates and tubes into the incubator in one of our upcoming videos. For now, you're good to turn off your burner and move on to whatever next step you might have. All right, so we're just going to give you a close-up of how the streak plate technique looks. So we're not going to bother showing you any of the flaming or any of those sorts of steps. We're just showing you how to go about inoculating your streak plate. There we go. So you can see we're filling in that first quadrant, and you can actually see fairly well that the loop has kind of the flat circular side touching down to the agar, so you're getting as much contact with that culture as you can. Now would be the time to flame, let it cool. We're accelerating this a little bit because we just want to show you the pattern here. And now you can see that Sarah's just using kind of just the top portion of the loop to make those extra lines, and she's being really cautious not to cross into that first quadrant again. Flame again. I always find it helpful to turn your plate as you go too. We're crossing through on those first 2 and then never touching the inoculated portion again. All right, and that's all it takes. So if I angle this up, you should be able to see all those lines that have been drawn. So when you go to do your first few streak plates, if you do even half that many lines, that's probably good. Just so that you're, not worried about accidentally crossing into the wrong zones. But once you get a little extra practice, you should be able to make patterns just like what you're seeing on this plate here, and this should hopefully get you to the point where you're seeing lots of isolated colonies over in this zone here after inoculation. So we have one more way that we want to show you as a possibility for growing microbes for this set of videos. and that is doing a pour plate. So right now Sarah's getting some prep done to get ready to do a pour plate. So she's opening our bag of plates and getting out a clean Petri plate. That's because in this technique you are pouring molten agar with bacteria in it into a plate. Sarah's making sure that that plate is completely intact. Often the first plate out of the sleeve ends up with some cracks or damage, and you don't want to have that accidentally spilling all over the bench. Now she's tightening up that sleeve, taping it shut so that those plates are clean for the next person. And now, before she goes and gets that agar, she wants to make sure that that plate is labeled. And remember which side we're labeling on. We're labeling on that kind of smaller diameter portion of the plate. And here you want to include your name or your initials, the date, the type of media that you're using, and the organism that you're putting in there. So today we're putting E. coli into TSA agar. The bench top has already been cleaned. Sarah's got her culture ready to go. Burner's nice and close. Everything is set and ready. And she's grabbed a piece of paper towel just because she has to get that molten agar out of the hot water bath. So she's gonna head there now and pick it up from the water bath over here. Can't quite see her getting it, but that's all right. And she's just wiping any extra water off of the outside of the tube so that it doesn't end up dripping. So now she's got to work a little bit quickly because that agar is going to start to solidify soon. So she gets the flame going and right away sterilizes the loop so that it can start cooling. Remember, we're getting it nice and red hot so that it's totally sterilized. And once it's been sterilized, we're keeping it nice and close to the flame, no more than 12 inches away. Now we vortex our broth culture, make sure that it's well mixed. And then we use our standard technique to take from broth. So we remove the lid, we flame the mouth of the tube, we go into the tube with our loop and grab a little sample of liquid, flame the mouth again, lid back on, and now we switch over to our tube of molten agar. Remove the lid, flame it, put our sample of bacteria in there, and just gently stir it around. We do not want to make bubbles in our agar, so we don't want to stir too vigorously. We will flame off our loop because otherwise we would end up getting solid agar on it. So we make sure that that burns off. And now it's time to mix our sample. We're absolutely not going to use the vortex mixer. Instead, we're going to do figure eights back and forth on our bench top to just gently mix it without generating any bubbles. And now we're still within 12 inches of the flame. We remove that lid, flame the mouth, lift open the lid of the plate, and pour in. Just pour out the entire amount, then put the lid back on, and set it aside. You can notice there that Sarah just did a couple little gentle back and forth, side to side motions. That just helps to make sure that the agar has mixed, but she's not doing it too quickly because we don't want that agar to jump back onto the lid. So now that that is in there, it just needs to sit on the bench for, usually it's about 10 to 15 minutes before it solidifies. And remember, you'll be able to see that it's solid because it will be kind of white in color. And then we'll be ready to put it into the incubator. So always make sure that the flame is turned off at the end of the procedure before you go to leave the incubator. And we'll see you back for the next very short video, just walking our way over to the incubator. Now that we've done all of our aseptic transfers, we're ready to take our newly inoculated tubes and plates over to the incubator. Notice that Sarah grabbed a second test tube rack to transfer her tubes. We don't want to take an extra trip with our cultures. They don't need to come for a trip, and we definitely don't want to carry our tubes in our hands. Always put your tubes into a rack before moving them to the incubator. So now you can go with your tubes and your plates and make your way over to the incubator. These tubes are getting inoculated at 35 degrees Celsius, as are most of your incubations for this class. Now we can, with our plates, we always want to make sure that our agar is facing up. So Sarah, you'll need to turn that the other way around, and we can put that into the incubator. Do the same with that one. And then usually we'll already have a rack in there, so just put your tubes side by side to each other. Want to make sure that there's room for others to put their tubes in there too. Now we'll close both of the incubator doors, so there's an inner door and an outer door. And now this is set and ready to incubate for 24 to 48 hours, depending on the experiment. And that's all that we need. So next time we'll be coming back and you'll be able to see, hopefully, some lovely plates and tubes of bacteria. Hi everyone, we're back after 24 hours of incubation, and we're looking at the results from your aseptic transfer technique lab. So as a reminder, we inoculated 4 tubes. 2 of those tubes had liquid culture, and 2 of those had agar slants. And we also inoculated some plates. So if we look at our results here, we had some E. coli that we took off of a plate and inoculated into a TSB broth. So we're all labeled up there. And you can see here that if we kind of make sure that everything's all mixed up, we can see that this broth is now quite cloudy, which shows us that bacteria have grown in there. We also did a broth to broth transfer, and again, that one is quite cloudy. So if we put the 2 of them together, you can see that they grew approximately the same amount. We also did a couple inoculations onto TSA slants. And I'm just gonna, yeah, you should be able to see that there's a nice kind of white squiggly line. It's pretty dense because a lot of bacteria did get transferred, but we have some nice growth on the surface of our slant. And we have that in both conditions, so whether we incubated it from a broth culture or from a solid culture. We also did a pour plate where we put our, we took a little scoop out of our broth culture and mixed it up into some molten agar and poured it into the plate. You probably can't see from this side, but if I move it and take the lid off, you can kind of see that there's kind of a spotty texture on the surface of the plate, and that is all growth of E. coli. So we have a complete lawn of E. coli growing on here. We're not able to see individual colonies, so we would call this a bacterial lawn. We also did 2 different streak plates, and we have very different results for them. So we have one of them here and one of them here. And this is the one that has the results we would normally expect to see for a streak plate. So we can see that, I will open it and show you the other way as well, but this whole area where you did all that back and forth, moving around, that is just solid growth of bacteria. Then we have some lines of bacteria in the next section, And it's not until the third section that we begin to get individual colonies. I'll show you it this way. Get that out of the way. So you can see that we've actually got individual colonies in that last quadrant. So those are ones that we would be able to pick from to inoculate over to a new plate or a new tube, whatever it is that we need to do. Now if we look at this other streak plate here, we can see that it's just lines all over the plate. Show it to you this way as well. So it's just a series of lines and no individual colonies. The reason for this is that we didn't actually flame our loop in between quadrants. And this is one of the biggest mistakes that we see students making when they do a streak plate, is forgetting to flame their loop and then ending up that they're bringing way too much bacteria over into the next quadrant. Just a reminder to make sure that you've brushed up on your streak plate technique, and always remember to flame and cool your loop in between quadrants.